Hi, good day, everyone. In today's revision class, we'll be talking about introduction to amino acids. Yes, of course. We are talking about the protein. We are talking about protein structure or chemistry of macromolecules, chemistry of amino acid and proteins. Like we introduced during our first day. To be able to understand proteins, we must understand amino acids. Now, going back to the definition, what are proteins? We define proteins as a polymer of amino acids. So for we to be able to understand this polymer of amino acid, we have to understand the amino acid itself. Now, that brought us to the question of amino acid. What are amino acids? So we define amino acids as group of organic compound containing two functional groups, the amino group and the carboxyl group, produced by animals, plants, and bacteria. Now, going back to history, we made mention that these amino acids have been detected many years ago, where we mentioned that asparagine was the first to be discovered in 1806 and threonine was the last to be discovered in 1938. These amino acids were named based on the first substances they were isolated or based on some physical properties. We said glutamate comes from gluten, from wheat, tyrosine from cheese. The name cheese in wheat is tyros. Glycine, based on the fact that glycine is very sweet in taste. Now, apart from the fact that amino acids, they form the basic structure of a protein, amino acids also perform quite a number of functions, where we mention neurotransmitters, porphyrins involved in the hemoglobin that are involved in transport of oxygen by red blood cells, talk about nucleotides, talk about all these nucleotide bases, where we say in the next biochemistry course where you do metabolism, you will see how you synthesize this nucleotide, where some amino acids play important role in the synthesis of this stuff. So we said normally the biological system, you will have a naturally existing amino acid up to 300, but only 200 of them were found in the proteins. So only these 200, sorry, only these 20 were found in proteins. Them and their modified form, you can have a modified form of these 20 amino acids that are found in proteins. So basically, amino acids were found in proteins and they are linked by a type of bond we call a peptide bond. And we said amino acids linked to form a primary structure, which form a secondary structure, which form a tertiary structure, which form a quaternary structure, which we know commonly as protein. Now, the chemistry of this amino acid. What is the chemical nature of this amino acid? We said amino acid is made up of two groups, the amino group, the carboxyl group, the hydrogen, and the R group. The R group is also called a side chain. It is the side chain that made an amino acid to differ from each other because this R group differ in structure, in size, electric charges. These influence quite a number of properties of an amino acid, including solubility in water. Another thing we mentioned is that these amino acids or carbon in amino acids are numbered in two ways. They can be numbered using a Greek alphabet, or they can be numbered using an Aramaic numerals. If they are numbered using a Greek alphabet, we said the carbon through which all the carboxyl amino hydrogen and the R group were attached to is called an alpha carbon. Now, any subsequent carbon that comes from the R group is now regarded as beta, gamma, lambda, epsilon, etc., up to omega in varies. For the Arabic numerals, the numbering started with the carboxyl carbon, where the carboxyl carbon is carbon 1, and then the alpha carbon, or the carbon through which the carboxyl amino and the hydrogen, all the other four groups were attached to, is the carbon 2. Then carbon 3 will now come from the R group. That is an amino acid. Now, isomerism in amino acid. Isomerism is possible, as we explained, due to the presence of a particular carbon we call asymmetric carbon or chiral carbon. And we say an asymmetric carbon or chiral carbon is a carbon to which all the four groups that contributed to the building of the outermost shell of this carbon atom are different. So when you have a different group contributing to filling of the outermost orbital of carbon, this carbon is regarded as asymmetric carbon, of which the position of these substances around this carbon atom can actually change. And when they change, they can produce an isomer of that model, which may have a different chemical properties. Now, in the case of carbohydrate, we set the position of the hydroxyl group around a carbon, the chiral carbon, determine the isomer. In amino acid, we set the position of the amino group, determine the isomer of an amino acid. So if you have an amino group at the right, you have the amino acid. If you have it at the left, you have amino acid. All the amino acids have a chiral carbon with the exception of glycine. Glycine does not have a chiral carbon simply because the groups that contributed to building the outermost orbital of glycine or the carbon or the chiral carbon in glycine, you have two groups that are similar, which is hydrogen. So you have two hydrogen attached to the chiral carbon, while you have the carboxyl and the amino group. Still, that made that carbon to be non-chiral carbon. So glycine does not have a D and L isomer. Like we said, normally, in biological system, we have many amino acids, up to 300, but only 20 are found in proteins. And these 20 are the ones we are going to discuss. We are going to see their chemical structure and then see the physicochemical properties in the next revision class. But before then, these amino acids were given the 3-letter code for simplicity, as well as the 1-letter code. You see glycine as GLY, usually the 3-letter code given to all the amino acids are actually the first 3 letters. Most of the cases, but you can have some cases where they are not actually the first 3 letters. Like glutamine. Glutamine is GLN. Asparagine is ASN, which of course it is not the first 3 letters in those amino acids. In 1-letter code, in 1-letter code also, the letter could be the first letter of the amino acid, or it could entirely be a different letter. Example, phenylalanine, F, tryptophan, David, tyrosine, Y, asparagine, N, glutamine, Q, aspartic acid, D, glutamic acid, E, lysine, K, arginine, R. These are not the first letter in the names of these amino acids. The letters assigned to them as the 1-letter code. It is important to know the 3-letter code of all these amino acids as well as the 1-letter code of these amino acids. This question may come where we refer to a particular amino acid using the 3-letter code or even a 1-letter code. If you don't know the 1-letter code, now it is very difficult for you to be able to interpret. And the question talk less about thinking of the answer to that question. It is in code when you should do them. If you have any question, please pass your question to the classroom, and the classroom will pass the question to me. And the question will also go, the answer will go back in the same direction. Thank you very much.